Hello, my name is Ulrike Schlegel and I am an eBRAINS curation scientist. In this talk, I will first give a short introduction to the importance and benefits of good metadata organization for a neuroscience researcher. And then I will explain how eBRAINS organizes and integrates metadata to provide an easy and user-friendly service and to support the findability, accessibility, interoperability, and reusability of research data in eBRAINS. This video is part of a collection of videos developed by the EBRAINS Data and Knowledge team, in which we explain the steps of data curation in eBRAINS. In this course, we introduce a few essential steps of data sharing, and we highlight why these steps should be a focus of any researcher who wants to share their data as fairly as possible through eBRAINS. The previous session was about data organization. The next session focuses on how to write a data descriptor. And in this lesson, I discuss how to organize metadata. But what is metadata? Metadata is data about data. It contains all the information that is necessary to understand the data. So let's look at a simple example. Libraries use metadata to organize their books, and you have probably used metadata more frequently than you realize. To find a book in the library, you typically use a database and type in the title of the book of interest or the author who wrote it. Importantly, the list of metadata extends beyond the author, title, and year of publication. For example, the subject area here, neuroscience, is a term that is shared among many books that all discuss topics related to neuroscience. This common property allows you to find books that might be interesting to you, even though you didn't know they existed in the first place. Metadata is not only used in libraries to organize books, but can also be used by a repository like eBRAINS for the organization of data, models, and software. Metadata can consist of any type of information, but typically serves one of the following three purposes. Bibliographic and descriptive information is used for the discovery and identification of data. For example, the location of the data can be indicated by a digital object identifier, a DOI, for online materials, or a shelf number in a library for library books. Other useful descriptive information that can facilitate the finding of metadata might include the date and time of creation or publication and the authors of the data. Information about the source of the data and how they were generated are typically captured by the metadata about the data provenance. These metadata describe the process of how the data was created, typically starting with the subject and finishing with the data file. All the methods and techniques that were applied to that subject to create the data should be discussed in detail here. Lastly, we have contextual metadata. This is information about the context of the data. For example, how was the data validated and which software is required to use the data. Including metadata for these purposes has a number of benefits for you as a researcher. Rich metadata can be used to improve the development of automated tools for optimized processing and analysis of data. It improves the discoverability of the data, links data with common characteristics together, and highlights underlying relationships. It reduces misunderstandings of data specifications as well as protocols, and the metadata standardization also allows data to become more accessible and improves the usability within and between research groups. So let's go back to the library example and look at the different types of metadata that exist. For certain properties of metadata, there's only a limited number of values that can be assigned to them. For example, the year a book was published, the language it is written in, and the type of resource it is, are all very clear, but also at the same time general. This is not the only book that was written in English or published in 2019. In other words, these values can be shared among resources. These terms are controlled and are used to generate a controlled vocabulary, which enhances the interoperability of the data and can facilitate automation. Then there are metadata properties, such as the identifier of this book, that are very distinct, but they are also unique to this particular resource. There's only one book that can be found with this identifier. Lastly, there are metadata properties that do not have a particular format and are typically filled in with a more elaborate description. The miscellaneous notes are an example of this type of metadata. The terms are not controlled and descriptions are typically free text. As mentioned earlier, some of the metadata terms are controlled. Controlled vocabularies are also used for ontologies and ontology representations. But what is an ontology? An ontology describes a set of concepts in a subject area or domain, their properties, and the relationship between those concepts. We can illustrate this with an example. A Purkinje cell is a cerebellar neuron, but it's also an afferent neuron. Purkinje cells can be found in the Purkinje cell layer, which is a cerebellar cell layer. Cerebellar cell layers are part of the cerebellar cortex, which in turn is part of the cerebellum, which is a regional part of the brain. Even though the example used here broadly has a hierarchical representation, ontologies are commonly not hierarchical. For example, the cerebellum is also part of the metencephalon, which is, again, a part of the brain. And each of these terms used for this ontology are controlled and have a unique persistent identifier that can be used to find the standardized description that is specific to that term. In EBRAINS, we rely on metadata standards to curate data. Metadata standards are intended to establish a common understanding of the meaning or semantics of the data, to ensure correct and proper use and interpretation of the data by its owners and the users. The development of a metadata standard for the neuroscience field is very challenging because it needs to cover a wide range of research resulting in experimental or simulated data, computational models, and even software. However, most of them make use of similar terms and concepts and can therefore be standardized without losing the study-specific aspects. openMINDS was developed for this specific purpose. It captures common attributes of neuroscientific research products, allowing for the comparison of seemingly unrelated research products. And it makes it possible to integrate them into neuroscience graph databases, such as the EBRAINS Knowledge Graph. There are currently 3 metadata models released and 2 more in development. The released models cover the core elements of neuroscience research products, anatomical location of the data, and provide controlled terminologies. The upcoming extensions are focused on the provenance of data within specific fields of neurosciences, such as electrophysiology, for example. As research products, openMINDS currently supports datasets with data from human, animal, or simulated studies, computational models, software tools, as well as metadata and data models, such as openMINDS itself, or BITS, for example, is. So let's look at research subject as an example to explain how we can organize the metadata about the subject using key value pairs. A subject can be described in a number of ways. For example, by their species, their identifier, or their age. as we saw with the book example, certain properties can be restricted to a specific type or number of values. In this example, this is the case for species and age. These key value pairs are controlled. For example, the age of the animal could be one of the following values from this list. But the identifier that is given to this subject can be chosen completely freely. This type of basic metadata is typically known by the researcher and should be organized in this manner. Within openMINDS, a collection of metadata that covers a particular topic is known as a metadata schema. So let's look at a dataset as another example. It has a title and can include one or more specimens that were studied. But in this example, it's not a controlled term, but a link to the subject that we looked at before. So the schema links to another type of schema. This linking of multiple metadata schemas forms the basis of a metadata model, which can be used by graph databases such as the EBRAINS knowledge graph. But for the conceptual understanding of how metadata is connected, it is important that each instance containing the context, the actual metadata information, has unique identifiers that can be referred to as UUIDs. Such instances and how they are linked to each other are collected in the knowledge graph. An important advantage of linking data is that the data becomes more findable based on connected keywords, and the controlled metadata values make the data interoperable. So we can also look at a simplified graphical representation that is inspired by openMINDS to explain how metadata is connected in eBRAINS now. So let's start with one dataset with 4 subjects. If this were an electrophysiology dataset, the method that was used in this dataset could be in vitro whole cell patch clamp recordings, for example. The method or technique is further associated with a protocol which gives more specific details about the method. The method in combination with the protocol was applied to samples from each animal and generated a number of data files. In the example we use here, we get 4 data files, 1 per subject, and the collection of the files is stored in the file repository. All these elements together constitute a simple dataset. And the dataset is furthermore linked to 1 or more authors that contributed to its creation, and it's being assigned a DOI. So one of the great strengths of controlled and standardized metadata is that the terms can be shared between studies, linking them based on common factors. In this example here, 2 datasets generated by 2 different sets of authors could share the same technique. And we can furthermore extend the schematic with anatomical location information by relating the data to information from a reference brain atlas. Experimental data sets from, for example, human, mice, or rat can be anchored to their respective brain atlases in 2 ways. We can create a semantic link to the brain atlas, which means that the dataset is linked to a particular brain area. This way, datasets from different studies targeting the same brain area are connected. We can also create a spatial link by adding the coordinates of a specific location in the reference atlas space. This type of anchoring is particularly useful for a dataset with files that originate from a particular location within a brain area. So for example, electrode implantations or in vivo single-cell recordings. As mentioned before, metadata is the foundation of the EBRAINS knowledge graph. Over the years, the network of interconnected instances has expanded and the number of datasets in the knowledge graph has grown significantly. By interlinking data based on metadata, researchers can, for example, find datasets of interest in the knowledge graph using our search engine, which is called Knowledge Graph Search. So datasets can be found either by free text field search field, by searching within specific categories, such as datasets, models, softwares, or contributors, or by limiting the results to specific species, modalities, or techniques. And when you find what you need, you will get to a landing page of a data set. And here you can find all the metadata that is related to this data set. So you can find, for example, a short description of the data, related keywords, a link to the interactive atlas viewer, the data descriptor, and many more things. So thank you for your attention. Hopefully this lesson was useful for you and your team in understanding some of the aspects on how metadata is useful for the neuroscience community, especially when sharing data. And I urge you to have a look at the other lessons in our EBRAINS data sharing course series on the INCF training space. And let us know if you have any questions. If you wish to share your data on EBRAINS, please submit a curation request at ebrains.eu/share data. And this video was made possible by the EBRAINS Data and Knowledge team and the partnering institutions for storing and computing. If you're curious to know more, send us an email or follow us on social media. Thank you.